This slide here shows us what the specification says that you need to know for this section. So it's topic 3.4.7, but as I said at the start of the presentation, it directly links to the work we did on classification. So you've got to kind of keep those principles in mind. And when you do exam questions, you'll often find that there's a crossover between, there'll be some questions about general bits on classification, and then it'll link on to this, how we actually measure these differences and how we measure diversity. So this is what we'll be looking at, and hopefully when you've followed through the presentation, you'll be able to do some exam questions and actually apply your knowledge that you've developed. So our major advance in investigating diversity is the fact that we can now compare the DNA-based sequence of any organism pretty much that we want to. Now, this lovely little picture here actually represents just a very small section of DNA-based sequence that has been identified and is being displayed as a result of a bit of fancy computer programming. But basically what I could do is I could take the information in your genome from the nucleus of one of your body cells, and I could create and map out the DNA-based sequence of all 3 billion DNA-based pairs that you have making up your genome. And I could take yours and I could make a direct comparison, doing a bit of computer programming, and compare yours and mine and see how similar we were to each other and how different we were to each other. You don't need to know about the exact kind of technique behind how they produce this, but basically each of those different coloured bands that you can see, and you can see kind of green, red, blue and yellow little bands, each of those is representing like a little fluorescent dye or a little fluorescent tag that is possible to tag onto our bases, so onto our adenine, guanine, thymine and cytosine. And as a result of that, and using computer modelling, you then can produce this coloured banding pattern. So obviously just looking at it like that, it would be impossible for us to read just using our human eye. So that image there is then scanned using lasers, and you can make a comparison and do all sorts of interesting stuff with it. So it's been a massive advance and has allowed us to be able to compare the DNA-based sequences of organisms. So by following through this method of kind of taking a DNA sample from either members of the same species or members of 2 different species and producing this DNA sequence and analysing it by basically by comparing this colour banding pattern that we can see these similarities and these differences and think about diversity. And it might surprise you, or you might already know this, to think that actually 98% of the genes we have, chimpanzees will have those genes, and in fact, we might be okay with accepting that, because we've learnt for a long time that those primates are going to be quite closely related to us in terms of evolution, and so we're okay with that. But then if you take something like a mouse, a mouse will share 92% of the genes that you have. Now that might be a bit more like, ooh, that's a bit strange. And then actually, 18% of the genes you have, actually an E. coli bacterium will also have those genes. So it gives us a really kind of strong sort of indication about evolutionary relatedness and evolutionary history, and that whole idea that actually way back in evolutionary history, 3,500 million years ago, we have common ancestors. So it's really quite fascinating. The other thing that it's worth kind of pointing out though, so although we've obviously kind of got all of this sort of DNA-based sequence that we're thinking, oh yeah, well we've got that in common, we have to recognise that although we'll have those genes in common, we know just from the work that we've done on slight changes within the DNA-based sequence to produce a different allele can actually have a consequence to produce a quite different protein. And so these slight variations actually are the thing that gives rise to this massive diversity that we can see. Let's just for a minute then think about how we can use this information from the DNA-based sequence to determine the phylogenetic relationship between organisms. Now if we think back to the work we did before, remember phylogenetic relationship tells us about basically how closely related organisms are to one another in terms of evolutionary history. Okay. Now what we will remember is when evolution takes place, basically what we're saying according to the theory is that one species gives rise to another. Now if we think about, obviously if one species gives rise to another, then initially the DNA of the new species will be really quite similar to the DNA of the original species. But over time we'll get more and more mutations in the DNA-based sequence of the new species occurring. And as a result of each of those different and those separate mutation events, we're going to give rise to more and more differences in that DNA-based sequence. And remember, more and more differences in DNA-based sequence means more and more different characteristics coming about because of those different proteins being coded for. Or remember, maybe a protein is not coded for as a result of that change in the DNA-based sequence. Now the thing that's really difficult to get your head around, and I've really still find this challenging to get my head around, is that these differences that are accumulating across thousands and thousands of years. Okay, and so obviously the more time that's passed, then the more time we've had for these mutation events to occur, and hence the more time we've had for these differences to arise. And so what we say by the end of it is that we share kind of less similar DNA-based sequence with more distantly related relatives in terms of evolutionary time, and more similar DNA-based sequence with those that we're more closely related to. Hence why, in terms of evolutionary history, you're going to have a more similar DNA-based sequence with members of the chimp family than you are with, if we compare ourselves to even if we think about comparing ourselves to other mammals, because we shared a common ancestor with them much further back in evolutionary history. So there's been more time for those differences coming about due to mutations, been more time for those differences to accumulate. It might help you just to get your head around it, to sort of see this kind of what could happen on a diagram. So if we have a look at the relationship we've got here, we've got chimpanzees, okay, and then here we've got us, Homo sapiens, and here we've got Homo neanderthalis, one of our kind of closest relatives in terms of other species of organism. So here on this axis here, we can see we've got going back in evolutionary history millions of years ago. And we can see from the diagram that about 5.2 million years ago, about here, we can see the evolutionary ancestor, where basically a common ancestor between the Homo sapiens and the chimpanzees. This is where the divergence of those two groups sort of separated off. So in this space here, between just over 5 million years ago and the present day, this is where all of those differences in the DNA-based sequence have been occurring, all of those mutations that have given rise to the differences in the DNA-based sequence that we see between chimpanzees and humans today. Now, we said from earlier on in the video, didn't we, that we had 98% of our genes in common. So you could almost say, oh, we only got a 2% difference. Well, if we think about those differences, and the differences even within the 98% of the genes we have in common, that's all to do with these little mutations in the DNA-based sequence that have arisen over this time period of 5 million years. So if we then think about all the other organisms that we're different to, it works in the same principle. Okay. So little bit of revision from the other unit we can remind ourselves of here. So remember, Homo sapiens is our species name, okay, and there's Homo neanderthalis and Homo erectus. So we recognise, and Homo habilis. So we recognise that obviously these guys here we're all separate species, but we recognise that we're all part of the same genera. Because remember, in our species name, the first part recognises which genus we belong to, and we can tell from here that we've all part of the same genus. Anyway, let's move on. So let's now see if we can use some information on some DNA-based sequences to actually establish some phylogenetic relationships between different organisms. What you've got on this diagram just here, just let me grab my little pen, is information about a DNA-based sequence in part of a gene of some different species of dolphins and whales. So we've got a fin whale, a humpback whale, a sperm whale, a bottlenose dolphin and a harbour porpoise. Now, we know actually that these organisms are fairly closely related in terms of evolutionary history, but what we're going to do is try and establish which ones are more closely related to others by looking at their DNA-based sequence. So using this information, what I'd like you to do is you should have it printed out on your PowerPoint, use it to complete the table on the next slide, where basically you need to identify how many of the nucleotides they have in common, and then use that information to establish which of these species is more closely related to one another and see if you can construct a phylogenetic diagram. So pause the video, use the bits of information to complete your table, and then see if you can work it out. Just in case you're not quite sure how to fill out this table. So for example, if I wanted to compare perhaps a humpback whale with a bottlenose dolphin, I'd look at my DNA-based sequence and I'd recognise that actually they have 43 of the bases in exactly the same position as each other. So I'll put 43 in there. So most of them are done for you. You just need to work it out for a sperm whale and a fin whale. So I'll let you pause the video, have a go at that, see if you can have a go at these questions, and then see if you can construct a phylogenetic tree. Okay, so hopefully in your table you've managed to work out that the humpback whale and the fin whale have 51. Oops, let's see if I get my pen in the right place. 51, okay, and the sperm whale and the humpback whale, they have got 50. So in terms of the question, which species are more closely related to one another, what you do is look at the information and go, well, actually, from what I can see, it appears the humpback whale and the sperm whale are more closely related to one another, and then the fin whale with the sperm whale. So the whales are more closely related, and then they're more distantly related to the dolphins, and kind of even more distantly related to the harbour porpoise. So we can tell that, the how do you know bit comes from the fact about that numbers of the DNA-based sequence that they share in a common position. So what we've got to remember is that the changes in DNA-based sequence that occurred due to mutations are what gave rise to those differences, that eventually when we get so many differences accumulating, we get different species coming about. So let's see if you can have a go at that phylogenetic tree. Okay, so hopefully you've managed to construct a phylogenetic tree that looks something like this. And you've got to obviously be able to explain how you came to your conclusions and how you put this together. So hopefully you've been able to see that the fin whale and the humpback whale are most closely related with their DNA. Okay, and because they're most closely related with their DNA, they're likely to share a recent common ancestor. And we can also see that the bottlenose dolphin and the harbour porpoise are also very closely related. So they share a recent common ancestor. So you show this by having these parts kind of here and here. Excuse my handwriting, I don't seem to be able to get to grips with using my finger carefully to draw the words. And then the sperm whale is obviously more closely related to both the fin whale and the humpback whale than it is to the bottlenose dolphin and the harbour porpoise. So it shares a common ancestor more recently with them here, whereas in terms of evolutionary history we have to trace it back to here, where we'd find a common ancestor with the bottlenose and the harbour porpoise. So obviously we can see how useful knowing about DNA-based sequences for establishing those relationships. Let's take a quick check back to the specification then and see where we are. So hopefully you're happy with the fact that we can investigate genetic diversity by looking at measurable, or observable characteristics. So looking at these things that we can directly measure, or we can easily look at the DNA-based sequence. What we're going to think about now is, why is the mRNA-based sequence also a useful way and a useful sort of measure that can tell us about the diversity and relationships between organisms.